So thank you, Sam. So good afternoon, everyone. And I would like to thank Paolo specifically and the organization team for putting this excellent program that really focuses on spatial transcriptomics. So I'm Nachiket Kashikar, and in the next, let's say 15 to 20 minutes, I will tell you about a new molecular cartography platform that Resolve Biosciences has launched. So you might wonder where on earth Resolve is. That is what are our spatial coordinates. So before you open, let's say Google Maps, here is a piece of information. So Resolve is headquartered in Monheim, Germany, sandwiched between Cologne and Düsseldorf. So the point of the matter is you now have kind of a map in front of your eyes, enabling you to visualize from where I'm talking to you. It is the same for us biologists here. Why, what, and where. And that leads us to cartography. So cartography is the study of and practice of making and using maps. So in a sense, it combines science, technology, and aesthetics. Cartography models reality in ways that communicate spatial information effectively. And at Resolve, we are those cartographers that produce molecular cartography of biological systems. Specifically, we spatially map and quantify the expression of mRNA transcripts at cellular and subcellular resolution at exquisitely high sensitivity, which is quantifiable, and in a highly multiplex manner. So be it zebrafish retina, which you see on this slide here, for example, let me get my pointer up and running. So be it zebrafish retina, be it plant system, or be it zebrafish brain, be it rather rare model organisms such as reptilian brain. So we can work with all that. So with cartography, essentially, we are not limited to any specific sample type. So we are looking into mouse brain, rats, organoids, and so on. Today, I would like to dive deep in some of the key applications where our technology is making significant impact. And we are going to start that with neuroscience. What you are seeing here is mouse brain. On the right half of the image, you see the coronal section of the mouse brain, and it's from the Allen Brain Atlas, reference atlas, which helps you to identify different regions of the brain, such as cortex, hippocampus, and so on. On the left side, what you see is about half of the hemisphere on which we perform molecular cartography using a panel of 100 genes. So now we are going to focus on the cortex. If the slide moves, give me a second, please. Right, here we are. So in the mammalian cortex, neurons and glia form kind of a pattern structure across 6 different layers, whose complex cytoarchitectonic arrangement likely contributes to cognition and so on. Here, what I'm showing you are 13 known laminar molecular markers, and we see these markers beautifully distributed across the layers. As a case in point, calbindin 1, so what you see in red, that is showing dense labeling in layers 1, 2, 3, and lighter labeling in superficial layer 5. If you look at the cells labeled in dark red, as I said. So what you'll realize is that the technology perfectly recapitulates that expression pattern. Now, to further highlight that exquisite specificity and sensitivity, we compare it to some gold standards. We compare the specificity of our data to the transcriptomic atlas published by the lab of Chris Ponting. So that's something that you show in the top left panel here. In the panel at this top, calibrated layer enrichment probabilities for chosen 5 classical layer markers are represented to the right of the corresponding in situ hybridization that you see here. Boxes outlined in black indicate that the classifier predicted the gene to be enriched in that layer, and the blue bars beneath, essentially, for each image, represent the highest expression level of that gene in any sample. In the bottom left panel, we show the expression pattern of exactly the same genes using molecular cartography. Please remember that in this particular experiment, we did not have those genes done at one at a time, but in a highly multiplex manner. Now, if we just take the example of CUX1 on the left side, what you see, we recapitulate exactly the same expression pattern as with ISH. The same is for CUX2, and the same is, let's say, for CTGF, which is in the thin layer 6B of the cortex. So this demonstrates really high spatial specificity that our technology affords to researchers. Importantly, our data strongly correlates with bulk RNA-seq. Here we have the correlation of correlation with R square of about 0.9. So furthermore, we are quantitatively at par with conventional single molecule fish. So I have not shown the data here, but I'm happy to discuss afterwards. So just to elaborate on that, so if you, for example, do single molecule fish, let's say in a tissue sample or in a cell culture, if you are going to observe 100 copies of a given gene in a given cell, we are going to observe almost all of those 100 copies in that cell. So we taken together, what I'm saying is that molecular cartography is highly specific and ultra sensitive. Now we look at 35 genes from that panel of 100 genes and in the same cortical regions. So these 35 genes represent different markers for different cell types found in the brain. What we capture here are different cells such as glutamatergic neurons in red, inhibitory neurons in yellow, astrocytes in magenta, and so on. Here we focus on a specific cell type, the inhibitory neuron. So that's my favorite neuron, my favorite cell type in the brain. It constitutes a small but a crucial neuronal class in the cortex. So while these cells comprise only 10 to 20% of the neuronal population, their connectivity and recruitment is essential for brain function. What we beautifully capture are distinct subtypes of inhibitory neurons. So for example, you see GAD1 positive cells in red, parvalbumin positive cells in green, somatostatin positive cells in magenta, VIP, which are rare, in yellow, and so on. If you use some of the capture-based technologies or sequencing-based technologies, probably you're going to average it over a large space. So you will not be able to distinguish between neighboring cell types, which you can beautifully do here. Now, for connoisseurs of neurobiology, you can also see beautiful expression of some of the transcripts in the processes. So again, the whole point is that using this approach, we are seeing really high specificity and high sensitivity to detect rarest of the rare cell types. Now, because of the quantitative nature, so each tiny diffraction limited that you observed in the images so far is one mRNA molecule. So we allow the researchers to perform additional data analysis by getting cell by gene and gene by cell matrix. So to this end, typically we use DAPI as a proxy for cell segmentation. Of course, there are multiple ways to do so, and we are happy to discuss that afterwards. What we have done here for the sake of simplicity and argument, let's focus on DAPI. So we show segmentation performed using QPath. We have here thousands of cells, and you can get to statistics such as signals per cell or spots per cell or detected genes per cell. Now, post-segmentation, we perform dimensionality reduction using UMAP. So analysis performed using CURAT, for example, allows for UMAP of cells measured by molecular cartography and is colored based on cell subclasses. The heat map shows that the transcripts for respective clusters are specific and clearly allow to identify key cell subtypes. The heat map is arranged by the size of the clusters and markers that are significantly enriched in these clusters. The GIF on the right side displays projections of such clusters back onto the tissue with corresponding colors. Here we visualize spatially segregated cell subtypes. For example, for glutamatergic neurons, we have 11 different clusters. You might appreciate different population of neurons in cortex and part of the hippocampus that you see in this image. And as has been displayed on this slide, you can also do the same for other cell types. Importantly, now you can also quantify the number or the fraction of different cell types. For example, here you have 53% glutamatergic neurons, 8% GABAergic neurons, and so on. Of course, the key significance here is that you get this information in full spatial glory. Now, we have implemented this technology in the context of disease, so together with a pharmaceutical company. So what I'm showing here is an example in the context of Alzheimer's disease using a particular mouse model called PPS1. So the two key questions we are asking here are how do amyloid plaques, the major neuropathology, specifically or spatially affect cell distribution, and how does the expression of key regulatory gene changes as a function of distance from the pathology? We specifically look into 4 signaling pathways: complement, inflammasome, Wnt signaling, and microglial signaling. If you take a bird's eye view and focus your attention on the left side of the screen, what you immediately notice is that the number of neurons is significantly reduced in the APPS1 cortex compared to the wild type cortex. In addition, we can see the yellow blobs in the APPS1 cortex, which are sort of highlighted here, and they are labeled using a dye called PFTA after performing our molecular cartography. Now turn your attention to the right, and what you notice is that certain genes pseudocolored in red, they are significantly enriched in the wild type cortex, and certain genes pseudocolored in green are enriched in the APPS1 cortex. But biology has long ceased to be descriptive science, right? So we can now take advantage of the quantitative power. We looked into the expression of all 100 genes around the plaque pathology. Here again, we confirm the specificity of our data. On the left side of the screen is data obtained using RNAscope, where we see enhanced expression of 2 genes CST7 and TREM2 in close vicinity of the plaques. So plaques are again highlighted in yellow. RNAscope has, of course, two major problems. It is an amplification-based method, and hence the signals are blobs of uneven size, and you cannot really do quantification. And second, the multiplexing power is quite low. That's why where Resolve's molecular cartography technology helps you. We not only recapture exactly the same expression pattern, that is the enhanced expression of CST7 and TREM2 in the close vicinity of the plaques, but we can now start to quantify. So we took each plaque as a center. So we had about 635 plaques here across three different brain slices, and we drew concentric rings around it of equal area. And we counted the number of molecules for each gene in such rings. Now, examples of 4 such genes are shown in the bottom right figure. So CST7 and C1qa in the complement pathway are enriched around the plaque. And while CYT1 and TUBB3 show diametrically opposite behavior. That is, their expression is diminished around the plaques. Now, here we take advantage of that cell segmentation to better understand the cell type specific changes in gene expression while having now that spatial context. With about 33,000 cells, the far left panel shows UMAP co-clustering of wild type APPPS1 cell types with microglia, astrocytes, and neurons. And other cell types, of course, differentially clustered. The same plot is also displayed in color-coded clusters, highlighting the source of cells from wild type or model mice as so shown in the middle panel, and indicating the distance of cells to the nearest plaque as shown in the far right panel. That's very much bringing the power of spatial analysis to the fore. Because only a few marker genes allow for cell type identification in molecular cartography, we could compare the effect of amyloid pathology and the relative abundance of different cell types. So for example, we've observed that glutamatergic neurons are really diminished in the APPPS1, and on the contrary, you have the enrichment of microglia. The spatial analysis performed also told us that there is a 20 to 30 micron sphere of influence around the plaques, around which this change in the cell type let's say quantification takes place. Furthermore, we spatially detect the shifts in the populations of microglia and astrocytes. So for instance, we found that there are few disease-associated microglia in the wild type cortex. So for example, if you look at the dark green, so you hardly see any. However, in the APPPS1 model, there is a major switch from homeostatic to the disease-associated microglia, and those disease-associated microglia cluster really around the plaques, the green population. And a similar shift in the spatial distribution was observed in the case of astrocytes and reactive astrocytes. So for example, if you look at the magenta and the red guys in the wild type and the APPPS1 cortex, right? Now, changing tracks briefly. So here we are looking at mouse liver, right? So in collaboration with VIB TechWatch, we aimed at benchmarking the spatial expression of 100 genes in mouse liver across 3 different biological replicates. So that's what is shown here in the pictures, and the reproducibility across those replicates is quite strong, with R-square being close to 1. And we compared also this data to published RNA-seq data, and again we observed a very strong correlation of R-square being 9.97. Here we focused on about 10 representative genes, and as we move away from the blood vessel, these 10 genes display a very peculiar expression pattern. For example, VWF and GATA4 closely line along the walls of the blood vessel, while CYP2E1, which is exclusively expressed in hepatocytes surrounding branches of the hepatic central vein, with high and low expression of this gene as we move away from the central vein. We compared again the expression of these 2 genes to published, and also specifically GLUL to publish conventional single channel SM fish coming from Ido Amit's lab. As I showed you with the brain data set, we find high spatial specificity in liver as well with molecular cartography. If you look at SM fish data around the central vein, we see 2 genes. GLUL1 is 1 cell layer thick and CYP2E1 is around 10 to cell layers thick. So our technology, with its multiplex approach, recapitulates exactly the same expression pattern with much superior capability towards quantification. Now here I'm showing you an example of cell type specific markers for liver. As a part of the portal triad, we see portal vein, bile ductules. We beautifully recaptured cholangiocytes solely based on the colocalized expression of 3 genes. Abundance of one cell type does not hamper the detection of rare genes or rare cell types. For example, even in the sea of hepatocytes here, we observe relatively sparse expressed cells such as Kupffer cells and cells of hematopoietic lineage. Now, of note, we looked into the subtypes of hepatocytes, and clustering showed that different types of hepatocytes are spatially distributed throughout the liver tissue. In particular, different subtypes surround the portal vein and the central vein. Such a differential spatial expression points towards differential function of different subtypes of hepatocytes. Having shown you the power of technology to uncover, visualize, and quantify so far hidden information in neuroscience and hepatology, so I'm going to show you just briefly some examples in oncology. So I'm not going to go into detail, but to better understand basic tumor biology or for better diagnostics or therapeutics, we need to understand the tumor microenvironment, that single cell resolution. And that's what I'm illustrating here. So what we can distinguish between different tumors obtained from these two different patients, patient 1 and patient 2. And again, you can dive deeper into single cell and subcellular aspects of the tumor microenvironment. So whether you're looking at neurology, oncology, developmental biology, or infectious diseases, the central underlying theme is we need to understand that tissue environment, microenvironment, to better understand cell-to-cell interactions and gene interactions and cell fates and so on. And we provide that opportunity. So let's turn our attention to the technology itself in the last 5 minutes. So how does it work? What are its features and why should you use it? So we have 3 unique differentiations based on 3 different factors. Number 1, high sensitivity, and I convinced you with some of the data, and high specificity. So, and second is exquisite resolution that allows to identify different cell types and also subcellular resolution. And number 3 is tissue integrity. So that is, even after performing molecular cartography on a given tissue sample, the tissue remains perfectly intact. That allows for additional dye labeling or any immunohistochemistry using antibodies. So how does it work? So to briefly explain, we typically start with a cryosection and even cell culture for that matter, adhere to glass. We design gene-specific probes using Resolve's own probe design algorithm that ensures highest specificity and fidelity of signal detection. The choice of genes is fully customizable. We hybridize the gene-specific probes to the tissue of interest. We color the gene-specific probes using fluorescent molecules through proprietary, unique, and patent file combinatorial coding process. We image the samples throughout the 10-micron thick tissue. Then we decolorize the probes, and this cycle of colorization, imaging, and decolorization is repeated a few times, and all this happens in a fully automated device without any user intervention. So what are the unique features that Resolve's molecular cartography technology brings to the table? So towards transformative differentiation and to provide unique opportunity to you. So we provide high multiplexing with up to 100 genes, as I said, and that number can be scaled up to several hundred genes. The technology allows for exquisite cellular and subcellular resolution. Importantly, we had 2-dimensional and 3-dimensional information through the thickness of the tissue section. Of course, typically we present that information in 3 dimensions. However, if you want that to make use of that 3-dimensional information in that 10-micron thick tissue, that information is readily available. Of course, it's an imaging-based technology, and you are going to generate lots and lots of data. And to really help you to make sense of that data, we have end-to-end bioinformatics pipeline to really analyze, to perform image analysis, transcript detection, co-localization analysis, and so on. And so it's a really interactive software. Importantly, it's digital quantification. So each spot that you see on the image is one transcript. As I alluded to before, so multiplexing, so we can easily combine the technology with post-hoc cell labeling using dyes or immunohistochemistry. Another key feature is throughput. So we can run up to 24 samples in parallel. And for each of those 24 samples, you can have a panel of those same 100 genes, or you can have different 100 genes in each of those wells. Importantly, we do not use any enzymes. There is no amplification or no capturing. So we have completely moved away from those stochastic and processes which might reduce sensitivity. And when it comes to sensitivity, as I told you before, it really matches the gold standard, that is conventional single molecule fish. Importantly, we are really flexible in the species that you can work with: mouse, rat, humans, plus zebrafish, Drosophila, and so on, different tissue types, and also different cell culture systems. So to end, so Resolve is at the forefront of the spatial transcriptomic revolution. We have just concluded our oversubscribed early access program and are launching full commercial service in Europe, including the UK, to enable researchers to answer their most exciting scientific questions, whether it is for basic science, translational science, coming from academics or pharmaceutical industry. We are also launching the full commercial service from our San Jose facility in the US in the coming weeks. And later this year, we shall expand to APAC. We look forward to talking to you. Please visit our website, www.results-biosciences.com. Follow us on Twitter for updates and exciting news from Resolve. We are actively recruiting. So folks in the audience who are in the job market, do check our LinkedIn page for some exciting opportunities in a fast-paced startup. Thank you.